All right, now that we've introduced the historical context of neuropsychology and the action potential, we'll spend the last few minutes to actually talk of methodology. Of course, hours could be spent investigating in details the functioning of the many neuroimaging tools that researchers can use to see inside the brain. But since this is a course on perception, and also possibly because I'm a little bit lazy, I'll only be explaining a few of them. The most important thing to know about the different methods is that the nature of the signal they use limits the quality of the recordings in either the temporal or spatial resolution. The temporal resolution corresponds to the accuracy with which researchers can infer the timing of the signals, whereas the spatial resolution, as you can imagine, corresponds to the accuracy of the activation map. Because every neuroimaging method uses a different signal, they each have a different resolution profile on those two dimensions. But a fundamental facet of neuroimaging is the statistical analysis that decide what activation is significant and what is considered noise. By far the most used research method in neuropsychology is the event related potential. It is based on the electroencephalography technology developed in the first half of the 20th century. The typical EEG has between 40 and 50 small electrodes that are distributed on the scalp of a participant that measure his electric potentials. Researchers can then expose the subject to a task or an event and correlate the brain activity with the cognitive processes thought to be implicated in it. It is important to note that since the electrodes are put on the scalp of the subjects and not inside the neurons, the primary electric signal cannot be directly detected and is thus inferred with predictive models. As electric signals travel via the path of least resistance in a brain with a highly irregular density, this prediction is not always accurate, and that gives the ERP its rather low spatial resolution. In other words, the individual electrodes do not necessarily receive the signal from their closest brain area, and that results in an imprecise activation map. An alternative method is to use the magnetic signal emerging from the electric activity that isn't biased by the path of least resistance. However, the has the drawbacks of giving a weaker signal that can't be detected from the deeper areas of the brain, and of being a costlier technology at that. That can be an important downside, as the reason why ERPs are so widespread is because the equipment is cheap and relatively easy to move around or transport to a patient's home. Fortunately, ERPs have an excellent temporal resolution, as the electric response happens quickly after the stimulation, which makes it the best method to examine temporal components of cognitive processes. This is a very active field of research. You see, every cognitive process can be decomposed into its electric components. For example, the presentation of a visual stimulus leads first to a negative charge in the 65 to 90 millisecond range in the primary visual cortex, and then to a positive charge at around 100 millisecond in the extrastriate cortex. The earliest imaging methods permitting a better spatial resolution of the brain was in the 70s with the computed tomographic scan, which involves taking slices of X rays to reproduce a 3D model of the brain's tissue's density. Simultaneously, the development of the magnetic resonance imagery in the 70s and 80s led to an even sharper spatial resolution, but at a much higher cost. The reason the MRI technology became so important then is because, contrarily to the CT scan, it permits an analysis of the activity of the brain, like the ERP. This is then called the fMRI. The fMRI is to the MRI what ERP is to the EEG, a functional analysis and its underlying technology. The better spatial resolution of the fMRI comes from the fact that it is not based on the detection of the distorted electric signal or its faint magnetic counterpart, but rather on the robust and accurate metabolic effect of brain activity. Indeed, a few seconds after a neuron has been activated, an efflux of oxygen is sent to it that can be detected via a radiofrequency pulse. As you can imagine, since it's relying on a delayed proxy for the neuronal activation, and since it necessitates a few seconds to gather enough data to have a robust inference, the fMRI has a lower temporal resolution. That means you cannot use it for transitory events like an epileptic discharge. It's just not made for this. The nature of the tasks must also be simplified, as the participant is inside a rather tight tube and so they cannot move. The statistical analyses behind those imaging techniques are also very important and rather complex. First of all, researchers must present varying conditions and filter out the activity not related to the specific cognitive process studied. They must then be careful in determining a criterion for what activity is statistically significant. The thing is, with those tools, researchers have sudden access to a gold mine of information. But the sophisticated aura of neuroimaging, with the beautiful pictures of the brain it generates, must not eclipse actual scientific rigor. There have been many papers published in reputable magazines with improper statistical analysis. The type of analysis that would make a dead fish's brain show cognitive states. This is not a joke. In 2010, a study by Bennett and colleagues showed that you put a dead salmon in an fMRI and you apply to it the type of methodology used in around a quarter of the studies published at the time, and it looks like you've engineered the world's first zombie fish. That's not all. In 2015, a statistical bug was discovered in the computational model underlying the technology that might have invalidated more than 40,000 previous fMRI studies. Of course, statistical analyses behind any type of scientific study can always be erroneous, and researchers have occasionally cut corners, but neuroimaging seems to be a particularly appealing layer of paint to turn a flawed study into a publishable one. Those neuroimaging techniques show the effect of a task or perceptual stimulation on the brain. But in the first class, I have mentioned that exploring the causal relationship between the physiology and the perception is much more complicated than that because you have to go play directly inside the brain. Straight up removing parts of the brain is ethically more challenging than it used to be. And so, unless you're willing to do it on a monkey, then you're out of luck. Now, ethics aside, there is nothing wrong with using monkeys as a proxy for humans. Their visual system is similar to ours, and they can be trained to be subjected to rather complex studies, investigating, for instance, their color vision or object perception. Once their performance has been measured, the researchers can then remove a particular area of their brain and retest the performance to investigate the role played by the ablated area. That's one method to go about it, but fortunately, there is one neuropsychological tool called transcranial magnetic stimulation that permits to magnetically depolarize specific parts of the brain without opening the skull. And honestly, humans usually much prefer to participate in those experiments instead. I have myself participated in a TMS study once, and it felt a little bit like when the doctor magically contracts your leg by a small knock to the knee, except he's literally controlling the activity in your brain. It's pretty insane. They were like folding my fingers without my input. Of course, that's a motor output then. But you could do the same to depolarize neurons involved in a perceptual process and ask the subject what he is experiencing. You then have a pretty good idea of what the region you activated is responsible for. You can also use the refractory period following a complete depolarization. You guys remember that, right? To investigate what happens when that specific area is disabled for a brief period of time. So, with this, we have now explored the historical context behind the two sides of psychology. As a reminder, I have mentioned that we can see psychology as a coin, when one side concerned with the cognitive and emotional processes, and one with the underlying structures of the brain. Another allegory one could use is how these two sides would study a computer executing a task. What would the cognitivist, interest in the processes, do? Well, you would control alt delete and look directly at the task manager to see how the CPU, RAM, and storage are all affected by the task at hand. I mean, I'm sure you already have thought about the similarities between those computer processes and human reasoning, short term memory, and long term memory, respectively. Of course, humans don't typically have a practical task manager ready to be conjured. And so, for a long time, the cognitivists or pre cognitivists, like the psychophysics, would have to infer the processes by running different commands and analyzing what manifest behavior is happening on the screen. Oh, look, if I open 67 tabs on Chrome, I completely crash the system. That must be its absolute threshold. But then you have the new cool kids coming to the party with their fancy instruments, and they just straight up open up that case and look inside at all the parts and their wiring and where the electricity is going. If you reach 90 degrees Celsius when playing solitaire, there might be something wrong with your rig. That's the physiological approach. But in all cases, a methodological requirement is to present a simple task and to be rigorous in the controls and the statistical analysis. Well, on that note, I'm excited to say that we'll be able to properly start our inquiry of perception by exploring vision next time. As always, you can become a Patreon to support me and get hold of the written guide containing additional information and quizzes. Thank you, namaste, and good luck.